Hello, my name is Xiang-Yu Liu in the lab of Dr. Siran Kang at Harvard University and Joslin Diabetes Center. Today, I will present our work. Hepatic glutathione depletion ameliorates MESO-D through selective protein oxidation and inhibition of lipogenesis, which is recently published on JCI. Glutathione is one of the most abundant antioxidants in cells, and it is generally thought to protect the tissues from damage. Decrease the levels of circulating glutathione and increase the levels of reactive oxygen species, or ROS, have been reported in liver diseases, including metabolic dysfunction associated steatotic liver diseases, or MESO-D. Thus, it has been widely assumed that hepatic glutathione depletion is detrimental, leading to MESO-D progression. However, the evidence supporting this assumption remains incomplete. First, many studies have focused on circulating glutathione levels, or its constituent amino acid, which may not accurately reflect hepatic glutathione status. Second, the reported decrease in plasma glutathione levels among MESO-D patients has not been consistently reproduced across independent cohorts. Third, and most importantly, the relationship between decreased glutathione and MESO-D is primarily based on association rather than demonstrated causality. So, in this study, we ask a simple but very fundamental question. What happens to liver metabolism if we selectively deplete glutathione in liver? To address this, we developed a liver-specific model to deplete glutathione. To do this, we took advantage of the enzyme, TRK1, which degrades glutathione into 5-oxoproline and cysteinoglycine. To overexpress TRK1 in liver, we utilized an AAV system targeting hepatocytes in adult mice. This approach allows us to deplete glutathione in adult liver without developmental confounder. Importantly, this model achieves the depletion of both oxidized and reduced glutathione without causing overtoxicity. We then performed metabolic phenotyping to understand how glutathione depletion impacts liver metabolism. Surprisingly, we found that glutathione depletion improves liver metabolic health both in chow and high-fat diets. Instead of causing liver damage, under both diet conditions, mice with hepatic glutathione depletion showed improved insulin sensitivity, lower fasting glucose, and enhanced hepatic response to insulin. Furthermore, on the high-fat diet, glutathione depletion decreased the fat accumulation in liver. At the molecular level, glutathione depletion caused a strong suppression of lipogenic gene expression and pathways involved in lipogenesis, fat synthesis, and storage, including SREBP1C and its downstream targets under both chow and high-fat diet. This indicates that glutathione depletion does not accelerate MESO-D, but unexpectedly protects from it. We then asked what the molecular mechanism is that contributes to these protective effects during glutathione depletion. Since glutathione is important for maintaining the redox homeostasis of protein, we performed a global protein redoxome analysis in liver by differentially labeling reduced and oxidized cysteines. Interestingly, glutathione depletion did not cause widespread protein oxidative stress. Instead, we observed highly selective oxidation of specific proteins. One of the most striking targets was fatty acid synthase, or FASM, a key enzyme for lipid synthesis. Consistent with this, fatty acid synthase protein levels were strongly decreased in livers with glutathione depletion. However, its mRNA levels remained unchanged. This indicates a post-translational mechanism where glutathione depletion selectively oxidizes and destabilizes lipogenic enzyme. Finally, we showed that TRK1 is suppressed in human livers during the development of methyl D, and also during feeding condition in mice, which may contribute to increased lipogenesis in both conditions. Integrating these findings into a working model, we propose that glutathione depletion in liver leads to selective oxidation of key lipogenic enzymes and decreases the transcriptional levels of lipogenic genes, which both protect against methyl D. Overall, our study reveals that glutathione depletion is not detrimental to fatty liver diseases, but instead can be protective. These results also indicate that targeting redox pathways could be a new strategy to treat metabolic disease. Thank you so much.